Hello and welcome to the Ion Exchange Chromatography demonstration for BL3322 Basic Biochemistry Lab. We'll be preparing a column of Cephalose beads to which we will add a mixture of alcohol dehydrogenase and glucose oxidase, which should separate these proteins as they pass through the cephalose at different speeds. Firstly, the Eppendorf tubes, which we'll use for collection, should be labelled. Now, we'll pour some water into the column just to make sure it's flowing okay. So we'll just pour this in the plastic column. There's a filter disc at the bottom of this column just to stop the cephalose from escaping. So if we open this clip, we should get a gradual flow through. If you don't, then if you ask a demonstrator or one of the technicians, they'll come and help you. We'll just let that flow through. We'll take our cephalose mixture here and give it a mix up so it pours well. And then we'll pour it into the column down the outside of a pipette. I want to make sure that we don't get any bubbles in your cephalose. So liquid will go through, leaving a column of cephalose. There we go. So we'll let the liquid flow through. What you don't want to do is let the cephalose dry out, because then your proteins won't pass through well enough. It's not essential, but a filter disc can be applied to the top of the column, like so. This is to stop the column being disturbed when you're applying your sample. So it just gets pushed gently down, just so it's on top of the cephalose. I'll add this funnel to the top so we can add more liquid. I can see that a column's forming and the meniscus of the liquid is close to the top. So I'll just close it there because I don't want it to dry. So next we want to equilibrate this column of cephalose using your equilibration buffer that you made up previously. So 16 mils of this. And just let that run through. So once the meniscus of the equilibration buffer has reached the top of the column, we can apply our 1.5 ml protein sample. We'll just let that run in a bit. I'll add a little bit of the equilibration buffer. Once our sample has run into the column, we'll add a further centimetre of equilibration buffer just to make sure it's all nicely running. Once our sample has entered the Cephros column, close the clip and we can add the first dilution buffer, the medium salt buffer. Then want to start collecting samples, so put my tubes under, starting at tube 1, open the clip, it should just drip slowly into the tubes, so there's no need to panic, it won't all flow out. We'll collect full tubes, 1.5 mils, until the meniscus of this dilution buffer has reached the top of the column. Once the first dilution buffer has reached the top of the column and you've collected about 13 samples, you can then add the high salt dilution buffer. And close this and switch to sample 14. We'll just continue collecting these samples. Once they've been collected, you can check for the presence of proteins in each sample by measuring the absorbance at 280 nanometers on a UV spectrophotometer. The samples or fractions can then be stored in the fridge for use in your STS page and Western blot.